Okay, is my screen visible to everyone now?
Yes, sir.
Okay, well, let us begin our discussion now. In the last lecture, what we were dealing with was the two hypotheses that people have put forward regarding how these parasites, the leishmanial parasite, might get entry into your macrophages. Okay, so the latest hypothesis which is now accepted is about the Trojan horse hypothesis, in which your leishmanial parasite considers that polymorphonuclear neutrophil granulocyte PMN as a Trojan horse, and eventually they get entry into the macrophages and continue their life cycle. Well, this is what we have talked in the previous lecture. Okay, then there is an alternate theory regarding how these parasite might get entry into your macrophages. Okay, you might find this alternative theory in some older books which you might happen to refer. Okay, now according to this older hypothesis, what happens is that the entry of the promastigote, entry of the parasite in form of promastigote, begins when this promastigote uh, goes and make first contact with the macrophage. Soon after it makes the contact, the process of internalization begins with formation of those pseudopods, and the after this pseudopods completely engulf your parasite, it gets entry inside the macrophage, and it forms something called as the parasitophorus vacuole. Okay, so now once this parasite gains entry into the macrophage, what happens is that the lysosomes which are present in nearby vicinity of these parasitophorus vacuoles, they start to migrate towards it, and they start to fuse with the parasitophorus vacuole, and thus whatever content that is present in those lysosomes are poured into your parasitophorus vacuole. Okay, so but still, even though the content from a lysosome that is poured into your parasitophorus vacuole, somehow the parasite has no effect of that content. Otherwise, what should happen is that when the lysosome goes and binds to any vacuolar body, those vacuolar bodies are usually destroyed, but somehow these parasite withstand the content of these suicide bags. And next we would discuss how exactly the parasite would manage to remain inside the very harsh condition that is created in this parasitophorus vacuole after the lysosome pours its content into the parasitophorus vacuoles. Okay, so the major drawback of this hypothesis so far is that it cannot explain the very nature behind how these parasite gain entry into these macrophages without activating it. Okay, so this is one of the drawback of this theory, but people still somehow teach in this way as well. So I thought you should know both the way of how this parasite might get entry into the macrophages. Okay, so now let us continue our discussion further. uh, it becomes now interesting for us to know how parasite manages to remain alive and of course replicate in the parasite in that parasitophorus vacuole, even after the lysosomes would go on fusing to that parasitophorus vacuole and go on pouring their contents into that parasitophorus vacuole, and the parasite has no effect on it. So how do this parasite manage to sustain itself and complete its life cycle in such a harsh condition? That is an interesting fact to know. Well, to explain this thing, there are two type of mechanism that are proposed by scientific community that would explain how this parasite how this parasite is kept alive in those parasitiforous vacuole, even though it is surrounded by the content of those uh, lysosomes as such. Sorry, those lysosomal content. Okay, so the very first mechanism that is proposed revolves around something called as the oxidative mechanism. Okay, so well, let us first try to understand what happens normally when the microorganism gets entry into macrophages. Okay, so upon entry of any foreign particle or any invading organism into those macrophages, that results in a very rapid respiratory burst that eventually produce a lot of oxygen. Okay, so there are certain set of enzyme called as NADPH oxidases that would convert your oxygen into reactive oxygen species that are collectively formed of many chemical species like hydroxy free radicals, singlet oxygen, and their other reactive intermediate. So these reactive oxygen species, being very unstable and highly reactive, they would go and attach to the macromolecular components like proteins. They would go and attach to even a DNA molecule and interfere with their activity. They might go and attach to the membrane. When such thing happen, if such reactive oxygen species goes and attaches to membrane, the membrane becomes fragile, its integrity is compromised, and thus after action of those reactive oxygen species, such membranes are very amiable for destruction, for disintegration, even if they undergo any small osmotic shock or even small physical contact with other organelles also. So in this way, the pathogen is destroyed by this respiratory burst mechanism using the hydroxy free radicals. Okay, but now what happens in Leishmania, what these parasite do is that it expresses some iron containing superoxide dismutases that would convert your reactive and highly toxic hydroxy free radical and related superoxide radicals into its less toxic form called as hydrogen peroxide, as you can see over here in form of H2O2. H2O2 stands for hydrogen peroxide. तो ये लेशमानिया क्या करता है, जो कुछ भी हाइड्रोक्सी फ्री रेडिकल तैयार हो जाते हैं उन्हें उसको क्या करता है, इट कन्वर्ट्स देम इनटू लेस टॉक्सिक फॉर्म हाइड्रोजन peroxide. And there are another systems, well say for example in mammal, we again convert that hydrogen peroxide into its benign form, into its least you can say least टॉक्सिक फॉर्म, दैट इज वाटर, उसका कंप्लीट डिटॉक्सिफिकेशन वाटर में किया जाता है. मैमल्स डू दिस थिंग विद हेल्प ऑफ ग्लूटाथायोन सिस्टम, while these parasites have evolved a specific set of enzyme called as trypanothione system. तो मैमल्स में आपका हाइड्रोजन peroxide का कन्वर्जन वाटर में कन्वर्ट करने वाले एंजाइम होते हैं ग्लूटाथायोन सिस्टम में. On the similar line, in these parasite, they have evolved a different set of enzyme called as trypanothione system that do the same function of converting hydrogen peroxide into water. Now, since there are these two entirely different set of enzymes performing this action, therefore the enzymes that are involved in trypanothione system forms a very attractive drug target, and they are a very attractive source of therapeutics that are involved. So if you target these enzymes from this trypanothione system, eventually you would be able to keep the concentration of hydrogen peroxide for a longer time in the in your parasite, and thus the parasite may suffer from oxidative stress, and eventually it can die off. Okay, so anyhow, this is how Leishmania avoids the product from respiratory burst. There are other few two mechanism that are also proposed that present how these parasite might sustain itself in content of the lysosomal containing parasitiforous vacuole. Okay, so these mechanisms are included in non-oxidative mechanism. Okay, so one of the non-oxidative mechanism includes expression of polyporphyrins. Okay, so these host cell, that is in case of humans, the macrophages are known to express channel or pore forming molecules, which we call them as polyporphyrins. Okay, so now these polyporphyrins result in perforation of pathogen cell membrane. They would go and attach to the cellular membrane of this pathogen, and eventually result in formation of pores, and due to formation of pores, the content of the cells would ooze out of itself, and the pathogen would eventually die. But in order to counter attack such a defense mechanism, what Leishmania does is that it starts to express a lot of surface membrane glycoproteins, and due to abundance of that surface membrane glycoproteins, these polyporphyrins somehow don't get chance to go and make holes in the membrane of the parasite, and hence it enables itself to gain resistance against such polyporphyrin mechanism that host might mount. Okay, this is one of the mechanism. Another non-oxidative mechanism involved action of enzymes like arginase and urease. Okay, so as per this enzyme, what Leishmania parasite does is that it raises the pH in lysosome. Okay, so normally the lysosomal compartment are highly acidic in nature, and it has got its pH about or around 4.0. Okay, so most of the pathogenic proteins or most of the other pathogenic components are degraded at this condition. But somehow what happens is that this Leishmania has some another strategy up its sleeves that I'm going to explain you now. So what this parasite does is that it manages to secrete some ammonium ions that would eventually neutralize the acidic environment in phagolysosome, and thus it would enable that parasite to grow and flourish in the parasitiforous vacuole, even though it is very acidic in nature. Okay, so raising the pH stuff happens in mitochondria. Okay, so over there the proteins are degraded, and whatever Rg9 is formed, it is acted upon by Rg9. It is acted upon by the enzyme called as arginase, that is responsible for its hydrolysis, and this arginase would hydrolyze your Rg9 into urea and ornithine. So there are specific urea transporter present on the membrane of mitochondria that would bring that urea out of the mitochondria, and in the lysosome, or in what we call it as phagolysosome, this urea would be converted to ammonia using the enzyme called as urease. Okay, so now this ammonia has got a positive charge, which is then used to neutralize the acidic environment which is present in form of negative charge in phagolysosome. Okay, so in this way, this parasite expresses a certain enzyme and uses some transporter to neutralize the environment, to neutralize the acidic environment that is present in the phagolysosome, and thus it manages to remain alive in very harsh and very adverse condition in phagolysosomes. Okay, so now by whatever mechanism, anyone wants to ask something?
So one question.
Yes.
So in the ammonium production in the previous slide, जो प्रोटीन डिग्रेडेशन हो रहा है वो प्रोटीन का सोर्स क्या है सर, होस्ट प्रोटीन का?
आरजी9 तो मैक्सिमम प्रोटीन में रहता है ना.
वो सही है सर, पर वो मतलब वो जो प्रोटीन डिग्रेडेशन है मतलब,
हाँ.
अच्छा अच्छा हाँ राइट राइट, आई गॉट द क्वेश्चन, थैंक यू सर.
Okay, so once well, by whatever mechanism it might be, it somehow sustains itself in the phagolysosome. Okay, so now let us continue our discussion on how the life cycle continues further. Okay, so now in this parasitiforous vacuole, there is metamorphosis, or you can say the slender form of your parasite would eventually get converted into some another form, which we call it as now MST-goat. Okay, so now this MST-goat get attach itself to the parasitiforous vacuole membrane by their posterior end, and following their attachment, it starts to divide very rapidly. There are multiple divisions of these MST goats, and due to repeated cycle of these multiple division, enormous number of MST goats are now accumulated in parasitiforous vacuole, and thus your parasitiforous vacuole also grows in size. It occupies the entire cytoplasm of that macrophage, and eventually they burst out of that macrophages, and MST goat can now either infect new macrophages, or if they are present around, they can be sucked up by sandfly to continue its life cycle into the insect host. Okay, so this is how things happen in a case of Leishmania. Okay, so well, let us now continue our life cycle in how things happen in the sandfly as such. Okay, because your life cycle in human host completes in this way. Okay, so since its life cycle involve bursting of these macrophages, and these macrophages are one of the very key player in your immune system, and due to its frequent bursting, the number of macrophages might come down, number of immune cells that are responsible for controlling other infection might go down, and thus infection of this parasite are usually associated with another co-infection. Okay, so they lay a kind of opportunity for other opportunistic pathogens like bacteria and fungi to gain entry into the parasite. Why? Because the immune system has been already compromised because of the infection of these leishmanial parasite. राइट, तो लेशमानियल पैरासाइट के साथ में लेशमानियासिस के साथ में बाकी के भी को इन्फेक्शंस भी पेशेंट्स में पाए जाते हैं. ये क्यों होता है? क्योंकि इम्यून सिस्टम काफी कॉम्प्रोमाइज हो जाता है वो टाइम में. ओके, so now let us continue our discussion on how this parasite completes its life cycle in the sandfly or in that arthropod secondary host. Okay, so to explain you that, I have a wonderful video with you, video to show you that would explain the life cycle. Okay, now I will show you that video. Okay,
Okay, sir.
life cycle of Leishmania in the insect vector.
Okay, is the video audible and visible to everyone?
Yes, sir.
Okay, now enjoy this.
The life cycle of the Leishmania parasite begins when a female sandfly takes a blood meal of an infected vertebrate host and ingests blood containing infected macrophages with amastigote forms. Observe the amastigotes passing through the thoracic midgut to the abdominal midgut. In this new environment, amastigotes are grouped together, forming clusters or nest cells, which are enclosed by the peritrophic matrix that protects them from digestive enzymes. Thus, amastigotes transform into procyclic promastigotes. Note that the anterior portion of the peritrophic matrix breaks down, and parasites are released to the midgut epithelium of the insect. Observe that the promastigotes divide by binary fission and attach to the midgut epithelium of the insect. Note that this adhesion occurs predominantly through the flagellum. Observe that some parasites are taken out with the blood meal. When the parasites detach from the epithelium, the promastigotes migrate towards the stomadeal valve, localized in the anterior midgut, where they concentrate and reinitiate cell division. Observe how many promastigotes are accumulating due to the division process. Observe that parasites are responsible for the production and secretion of a gel that acts as a plug, obstructing the midgut and pharynx. During the production of the promastigote secretory gel, the parasites start to transform into infective metacyclic promastigote forms. This differentiation process is designated as metacyclogenesis. Observe that during metacyclogenesis, the promastigote cell body is reduced in size, the flagellum increases in length, and the parasites become highly mobile and infective. Metacyclic forms cause damage to the stomadeal valve, interfering with its function and facilitating reflux of parasites that move anteriorly into the proboscis. They are introduced into a new mammalian host when the sandfly bites again, starting the cycle over.
Okay, shall I play it once more?
Yes, sir.
Yes, sir.
Okay, here we go.
The life cycle of Leishmania in the insect vector. The life cycle of the Leishmania parasite begins when a female sandfly takes a blood meal of an infected vertebrate host and ingests blood containing infected macrophages with amastigote forms. Observe the amastigotes passing through the thoracic midgut to the abdominal midgut. In this new environment, amastigotes are grouped together, forming clusters or nest cells, which are enclosed by the peritrophic matrix that protects them from digestive enzymes. Thus, amastigotes transform into procyclic promastigotes. Note that the anterior portion of the peritrophic matrix breaks down, and parasites are released to the midgut epithelium of the insect. Observe that the promastigotes divide by binary fission and attach to the midgut epithelium of the insect. Note that this adhesion occurs predominantly through the flagellum. Observe that some parasites are taken out with the blood meal. When the parasites detach from the epithelium, the promastogotes migrate towards the stomadeal valve, localized in the anterior midgut, where they concentrate and reinitiate cell division. Observe how many promastogotes are accumulating due to the division process. Observe that parasites are responsible for the production and secretion of a gel that acts as a plug, obstructing the midgut and pharynx. During the production of the promastogote secretory gel, the parasites start to transform into infective metacyclic promastogote forms. This differentiation process is designated as metacyclogenesis. Observe that during metacyclogenesis, the promastogote cell body is reduced in size, the flagellum increases in length, and the parasites become highly mobile and infective. Metacyclic forms cause damage to the stomadeal valve, interfering with its function and facilitating reflux of parasites that move anteriorly into the proboscis. They are introduced into a new mammalian host when the sandfly bites again, starting the cycle over.
Okay, so everyone watched this video. सबने देखा ये वीडियो?
Yes, sir.
Yes,
It was
sir.
really
Okay.
interesting.
Okay. So now let us come back to our slides. Okay, this is what we have done, and over here this is the summary of what you have seen just now, and I would just summarize it. What happens or what are the events that take place in the fly host of this organism. Okay, so the life cycle of this leishmania begins, leishmanial parasite begins when the female sandfly takes a blood meal of an insected host. In our case, that was human. Okay, it ingests the blood containing macrophages and parasites in the MST-good form. Okay, the parasite then passes from thoracic midgut to abdominal midgut. In the abdominal gut, parasite lives in cluster, and those cluster of cells are called as nest cell. These clusters are surrounded by something called as peritrophic matrix, and this peritrophic matrix is responsible for protecting these parasites from the digestive enzymes that are present in the fly host abdomen or the fly host gut. Then these MST-goat now forms or they transform themselves into procyclic promastigote. The anterior portion towards the midgut of this peritrophic matrix now breaks down. The parasite now come out of that peritrophic matrix and enters in the midgut of that insect. In the midgut, they start to rapidly divide by binary fission, or what we call it as longitudinal binary fission. Is one of the form of asexual multiplication that this parasite possess. And once they get divided by this binary fission, they get attached to the midgut of that insect. The flagellum of the parasite so far helps them in the attachment of procyclic promastigote to the midgut of the epithelium. Then following this, some parasite that eventually fails to attach to the midgut wall are excreted out with the blood meal that is not digested. Now these attached promastigotes start to migrate towards the stomadeal wall. They get concentrated around the stomadeal wall and again initiate a rapid division. Parasite now produce and secrete a gel, what we call it as promiscuous secretory gel, and this gel act as a plug that obstruct the midgut and the pharynx. So eventually, when this promiscuous secretory gel is being secreted, and in those set of circumstances, your parasite again gets transformed into a very infective form called as metacyclic promiscuous form. This differentiation process is also sometimes termed as metacyclogenesis. In metacyclogenesis process, what happens is that the cell body of that parasite is reduced in size, and the length of the flagellum increases very rapidly, and due to this the parasite become highly mobile and it becomes highly infective. Thus, in this metacyclic form, it is responsible for causing a lot of damage to the stomatial valve and interfere with its normal functioning. So now the stomatial valve is damaged, the parasite can successfully get entry into the proboscis, and when such fly takes another blood meal, it gains entry into the mammalian host, and the subsequent life cycle continues in the mammalian host as such. Okay, so this is all about the life cycle of Leishmania in the fly host as well as in human host. Okay, so now is the time that you start asking the questions.
Sir,
Yes.
one vector of this Leishmania sandfly, they sprayed by this sandfly. And how about, is there any other genesis, any other vector that this Leishmania spray?
No, so far so good. These sandflies are one of the major cause of vector. I mean, they are the major carrier. Okay,
Okay,
so these are the known vectors that we know. There might be someone
okay,
else, but I'm not sure about that.
okay, okay. Thank you, sir.